Thank you so much. Well, it's a pleasure to be here. Uh, the lunch was so good. Actually, it was way too good. It was like the type of lunch, the type of food that goes with my diet, special diet, which is called seafood diet. And so it's a pleasure to be here. First of all, I would like to thank those who have helped organizing this event to share ideas. And let's talk biochar a little bit. Let's talk biochar on soybean growth and nodulation. I start with a little introduction. Here, well, in this group, of course, in this audience, we know about biochar more than the rest of the population. This issue has been covered by previous speakers here, so I don't devote too much time to it because I have plenty of slides to share with you and little time. The technology has been with us since antiquity for a long time, obviously. We know about biochar, and at the same time, we don't know about biochar. As you noticed, previous speakers again, for instance, if you have biochar and you go and apply your biochar to a mollisol, you probably don't see much of any impact. You don't see much of any difference. You go and now add that same biochar to a soil, for instance, alfisol or antisol. You see a bigger impact of biochar. To do justice to this issue of biochar, one has to know one's biochar thoroughly, and one has to know one's soil thoroughly. And those combinations sometimes don't come easily, and they can't put them together, but teamwork and collaboration is always helpful. All right. When you look at the literature, so you see statements that in some cases, biochar worked, and biochar did an amazingly, tremendously significant difference in plant growth. And then someone else comes and says, I don't know, we are not so sure about this issue of biochar, and we now have to see it in a different light because we did not get what we expected after applying biochar to our soils. So you have literatures, of course, randomly selected some of the statements made or conclusions made from here and there. What we did in this study was to look at the effect of biochar on soybean growth and nodulation, the symbiotic association of rhizobia with plant roots. We looked at the leaf area, leaf growth, the stems and pods, pod formation, plant height, root length, root weight. And I'm here to share part of that study with you here. What we did in a greenhouse experiment, the treatments are listed here as no biochar with no soybean, biochar, no biochar and soybean plant, and 2%, 5% of two different types of biochar from different feedstock precursors, Miscanthus gigantus, and we have the pine biochar also. Now I have data from another experiment, switchgrass that sometimes I combine that data to explain certain things and compare certain issues. Now, the soil was an alpha-sol cation exchange capacity, so the chemical properties of the soil had low P and enough K and calcium and magnesium. Greenhouse experiment we conducted when we decided to sample our soils at different stages of growth. And what we did at 1 day, 3 day, 10 day, 30 day, and 60 day, we sampled the soil. The reason we sampled the soil to track the activity of enzymes that I will share later on in this session with you. We added phosphorus to our soil because it was low. Along with that, we added some potassium also. Now, I would like to share some ways of producing biochar, because how you produce your biochar really impacts the properties of biochar and then the effects of biochar on plant growth. There are ways, of course, to produce biochar. We produce our biochar using the slow pyrolysis. And you have seen, most of you, this picture that when you introduce biomass into the pyrolyzer, pretty much more or less you get 50% of it as biofuel and liquid fuel, and then the 50% of carbon going into biochar, depending on many other factors, of course. And how do we produce our biochar? It's a very rudimentary system that we have here, and designed and built based on publicly available knowledge on internet, and adjusted to get the temperature we need. This consists of, basically it's mobile. We can move it from one place to another place easily. And it consists of two barrels, one the interior and then the exterior. And to overcome the activation energy we need to keep the thermochemical reactions going, we fill the exterior of the inner bin with biomass itself, mostly. And we ignite that, and then the biomass that is supposed to be being converted to biochar is the interior of this one. And then we produce our biochar. We track also the temperature, and temperature that we produce our biochar is between 400 degrees and 600 degrees. So most of them around 500 degrees. So this is the biochar that we have produced from different feedstocks. And what you see is like we also keep track of the temperature that they go through, the thermal history of biochar, that is. So, for instance, this refers to that thermal history. So you see the endothermic and exothermic reaction within the interior barrel. And this one is another type of biochar. I can hardly read it from here, but you can read it probably there. And this is another woody ligneous biochar we are producing from a tree, and this is another one yet, and that's another one. And then biochar, for instance, this one looks pretty much like miscanthus, producing from miscanthus. And then this is the pine biochar that we used in this study also. And then after the biochar is produced, we crush it and pass it through 1 centimeter sieve. And we mix it, mix fertilizer, and mix the biochar with the soil. And then, of course, then we use it in the greenhouse experiment. Biochar, as we all know, is recalcitrant for the most part, has sorption properties. Biochar is charcoal. Really, biochar, you all know that biochar is charcoal. In other words, you can burn it and you can make shish kebab using biochar or whatever. You know, I've heard biochar being used for remediation. I'll use it for remediation purposes also here and there with my research. And, but biochar can be used to make a hamburger. You know, actually there are people out there in the Middle East and southern Russia, places like that, that they use that biochar to make shish kebab. It is produced through pyrolysis, deprived oxygen environment, and these are the areas interested in my lab. Basically, we are using in the different aspects of biochar. Nutrient cycling is one of them, of course, and which has something to do with enzyme activities within the soil, and etc., etc., and as a soil amendment especially. Oh, now we're going to be talking about results in discussion. Of course, as you know, biochar is important in sequestration of CO2 from the air. That's always in the background, but you can't tell a farmer to go and use biochar because you want to sequester the CO2 from the air, because they're not going to pay attention to you at all. When you talk to a farmer, especially I talk to small farmers in my university, and you talk about, let's say, you say, why should I use biochar? Say, well, because you're going to benefit from it, because you're going to increase your yield. You're going to make money off of it. You have biomass here and there. You can actually change that biomass into biochar and sell it, perhaps. So let's talk about that biochar, some of the properties of that biochar. Yeah, there has been talk about pHs of biochar, and some researcher was seeking a biochar with low pH. If you want biochar for low pH, you use coniferous trees. You make your biochar for coniferous trees and has a lower pH, significantly lower pH. If you make your biochar from herbaceous biomass, you expect the pH to be high. So, as you see biochar, you can't say just I use biochar here in this state or that state. The results are not going to be comparable because your biochars are different and your soils are different. So you'd have to be able to compare things. You would have to have exactly the same biochar, prepared exactly the same way. All right, and the soil that you're using that biochar on ought to be the same. Otherwise, plants are going to grow differently. Otherwise, everything is going to be different. So, no surprise there. And also, biochars have nitrogen in them, you see. There are some biochars, herbaceous biochars, that can pass as a nitrogen fertilizer, for instance. And you see, you add biochar to some plant, it starts growing big, and you say, what happened? This is a miracle. I add charcoal, and for ages, speakers before me have talked about this, that they added charcoal and plants started growing. Even Justus von Liebig that we were talking about, law of minimum, remember one of the speakers talked about it. He knew about nitrogen, he knew about all this stuff, and he talked about charcoal char, biochar. And because biochars have nitrogen. Pine biochar, little nitrogen. Herbaceous biochars, more nitrogen. Switchgrass biochar, lots of nitrogen. All right? Okay. When you add biochar to your soil, your bark density comes down. This shows it very clearly. All right? Bark density, untreated here. And is there a way to control the lights in this room, or can you see it well? All right. This is important. I don't want you to miss this important information. All right. So 2% pine biochar. You see the bark density comes down, of course, you know. So you get aeration, so you get more oxygen in there, and water moves, hydraulic conductivity goes up and stuff. This is basic soil science stuff. And the soil pH, you add the biochar to this alpha soil, for instance. You know, what happens is that your pH goes from 5.5. With pine, doesn't make much of any difference, but then you go to herbaceous biochar, switchgrass, dump that data from some other experiment in there for you just as a bonus. And here you see that the pH goes up, okay? But you see the pine biochar did not increase that pH. Okay. Root growth of soybean. With here, the biochar here is pine. Visually, when it comes to root, you don't see much of anything, plus the fact that when you're talking about root length and root density in a pot, 5 kilograms of soil, what do you expect? Like plants trying to grow in a situation that is more like, no, really? I have to go very quickly then. Root length, you see that here, we had with that biochar increased significantly the length of the roots as opposed to this control. Root weight, you notice that also. Unfortunately, I don't have much of any time. And here you see the nodulation, you see the pine biochar at lower rate increase the nodulation, then no difference, and then you realize that when you use herbaceous biochar, the nodulation goes down significantly, okay? Again, that's because of the nitrogen, presence of nitrogen. You see that this is again biochar growing at different rates. Here in this case, we have pine biochar. And here you have the growth of soybeans in the presence of Miscanthus biochar. At different rates, zip through these quickly. The stem heights. You see that the leaf area of biochar increased significantly in the presence of Miscanthus biochar, as you can see right here. You see the pods. When pod biochar is present, pods increase significantly. You can see. Highly significantly increased. Developed pods, highly significantly increased. And the conclusion here, rate of the biochar significantly affects its ability to impact soybean growth. Root growth was significantly increased by application 2% biochar, but at 5% rate, root growth was reduced as indicated by the root dry weight. And the herbaceous biochar significantly inhibit nodulation in soybeans. Pine biochar significantly promoted nodulation at 2%, but then it dropped because of nitrogen. The effect of biochar on stem was influenced by the type of biochar. Biochar significantly increased the number of pods. The ability of biochar to enhance soybean growth is significantly affected by the type of biochar. Biochar as a soil amendment may contribute to sustainable use of fertilizers. I don't have much time, and I'm under fire here.
You're out of time. Sorry, Raymond.
You're out of time. So I'll see you later.
Yep. Raymond does have one more